So thanks all the jury member for choosing me for this exciting awards. And thanks IRCC for supporting me throughout all of my research career at IIT Bombay. So today what I'm going to talk about that way we talk, we able to think, we able to do everyday work. That means that billions of protein like nanoscale level, they are actually working for coordinated functions in our body. That's why we able to do all functions, whatever we able to do everyday life. So these proteins, they able to do functions because they are three-dimensional structure. So now, if there is any problem that to make that three-dimensional structure, what protein do, that most often they do aggregates and creates a disease. So one such of aggregates is example is like amyloid, and they're kind of specific form of these protein aggregates. So these are like, as you can see in the screen, this is one of the beautiful image of the amyloid aggregates. They are highly ordered cross beta CD structure, stable 2 to 50 nanometer in diameters. So the good part is that they are stable against pH, stable against proteases, high temperature. So they are probably one of the wonderful material ever evolved in nature, except they kill the cells and make a disease. So for example, these protein aggregates, protein aggregation creates almost 24 human diseases such as Alzheimer's, Parkinson, many aging related disorder, as well as type 2 diabetes, prion diseases. So here you can see that all these diseases, individual protein, they aggregates and form amyloid and they kill the cells, is as simple as it is. So then, as I told you, so since they are highly stable and they are kind of super material, people always wonder that is this material, protein aggregates evolve in nature, only creating human diseases? Impossible. So then people gradually able to discover that amyloid can also do a functions, native functions of the host organisms. So people able to discover that in E. coli, bacteria, they are abundant, functional amyloid. Also, many mammals, they also have functional amyloid. For example, one of my previous discovery when I was doing postdoc, we were able to show that protein peptides, they can form amyloid and store inside the secretory granules, and they're able to release the functional monomers. So that means there is an amazing kind of coordination of the protein. They can make misfold purposefully and aggregates, and again can do the functions when organisms need it. So then, when I joined at IIT Bombay, we hypothesized that since they are kind of one of the most robust material in nature, only problems they kill the cells. Can we evolve? Can we design such a way that we can make a amyloid utilize all their good functions without cellular toxicity? So we hypothesized that we can make amyloid-based kind of hydrogels for stem cell differentiation. We can use this amyloid hydrogel for drug delivery. We can even design for in vitro tumor modeling as well as biocatalysis. So a couple of examples I will show briefly. So if you want to design for tissue engineering, one of the things we have to remember that cells, they are kind of attached very nicely to extracellular matrix and make a tissue. So we wonder first that amyloid fibers have intrinsic capability to ordering cells, cell nicely adhere to the amyloids or not. So what we did, we took wide variety of amyloid fibers by protein peptides and first show that cells, irrespective of sequence, they're able to adhere nicely, they're able to spread, they are much more happy. So that after doing that, one of my students started designing peptides-based, amyloid-based biomaterial. So in this first work, we were able to show that we can make amyloid-based hydrogels. And if you put human mesenchymal stem cells, they actually differentiate into neurons. This is wonderful data in the sense that you don't need any growth factors. Itself, that amyloid nature of the hydrogels actually differentiating stem cell to neurons. So then, for the next study, what we did, that one such of the gels we introduced in the Parkinson brain using the GFP level mesenchymal stem cells. So inside the brain in the animals, we were able to see that how hydrogels actually contain that mesenchymal stem cell and actually helping this mesenchymal stem cell to differentiate into neurons. So then we are able to also show that if we are able to encapsulate growth factors inside the gels along with mesenchymal stem cell, we're able to show actually they are differentiating much more nicer, to the neuronal cells. So based on these, one of my students, he opened a company in San Francisco, and he's doing fantastic job and able to utilize these kind of materials, so that he's wondering whether we can use this material for the Parkinson disease therapeutics. So then another of my postdoc, what she did, that she was kind of imagining that can we utilize this material to kind of design, or we kind of make tumor inside the test tube. For example, as you know that individual cancer patient, they require different amount, different doses of drug or different types of drugs. So personalized medicine is very important. For example, that can we isolate the tumor tissue from the human, make the same tumor in the test tubes, test the drugs, and figure it out which drugs is effective, and go back to again to the humans and utilize those drugs so that personalized medicine could be effective. So for that, we wondered that can we mimic the tumor inside the test tubes, what is there in human body. So first, utilizing wide variety of cancer cells, we able to show that our amyloid hydrogels, they able to make tumors spheroid. Only problem is they are very heterogeneous in size. So therefore, the drug testing for each experiment will result different kind of amounts. So then what we did, we developed a new technology that called drop casting method. So basically using the cells and amyloid hydrogels, we make 96 OL plate where we put a single drop, and that drops actually gradually makes tumors spheroid, you can see. And those tumors spheroid, they are healthy cells also, mimicking the necrotic core that's supposed to be there in the solid tumor. So that means we are able to make fantastic tumor model using the different type of cancer cells. And you see that depending upon the gels, we can mimic the necrotic core as well. So that means different type of cancer we can actually develop in 96 OL plate. And using the RT-PCR, we are able to show that our gel even much better than whatever that famous hydrogels called matrigels. So then one of the questions asks that we use cell line so far. Can we use kind of, in vivo tumor and develop this tumor spheroid? So we develop the tumor inside the mouse, isolate the tumor tissue, and using the same technology, drop casting method in 96 OL plus, we are able to beautifully develop tumor spheroid. You can see how nice spheroid they develop and inside the necrotic core, and that is the characteristic of original tumor, solid tumor. And you can see we detail characterize these tumors, and we're able to show that original tumors and whatever we develop the tumor inside the test tubes, they are almost similar in terms of gene expression, in terms of their heterogeneity. So this is patents are filed, and US and PCT, as well as we are initiating that can we open up a company, and we are now trying to really look at that human samples in collaboration with actor. So another things that we are imagining, and that can we use this class of hydrogels for drug delivery applications. So different types of drugs we can encapsulate and use for the slow release mechanisms, so that these kind of drugs can be functional. So at first, so we took the different types of amyloid hydrogels, we design, and these hydrogel based on functional amyloid. So we make sure this amyloid cannot do the bad things to the human when you apply, because these are derived from the functional amyloid. So then we tried trans-retinoic acid, doxorubicin cancer drug, insulin, growth hormones, using a wide variety of small molecule protein peptide. We try to think that what kind of molecules actually these hydrogels can capable for the slow releases. So then one of the example I will show you for the insulin for diabetes treatments. So if we even implant inside the, let's say, subcutaneously in the mouse, we're able to see there is some inflammation at 7 days, but 14 days, all inflammation gone. And that is kind of natural for anything you implant inside the human or inside the animal. So we able to show that these materials are safe to use it. So then when we utilize with diabetic animal model, we're able to show that our amyloid hydrogels is fantastic up to 48 hours time scale to suppress that kind of glucose levels and to kind of mimic normal insulin secretion. So they're able to kind of mimic gradual insulin release. And now we are targeting, can you use once a week injection model using our amyloid hydrogels. So that's, so I will, so this is kind of our data. Thanks our entire teams, and also a lot of collaboration with Samir, Garindam, and different international collaboration, as well as Sudeep, Sadhana, Asutosh in our department, and a lot of our current members and past members of our team. And also thanks IIT Bombay to provide all the infrastructure and support, BIRAC and DST, DBT, all different kind of agency. And thank you very much. Again, I will be happy to take any questions. Yeah.